So we have briefly discussed that how important is the selection of secondary structural element to the field of protein engineering. But actually, when in practice, we don't select just an alpha helical or beta sheet structure. Rather, we end up selecting a super secondary structure. Now, what is a super secondary structure? So you can say that it's a three-dimensional protein structure which is composed of several secondary structures, but it's a bit smaller than subunit and um, and a domain. Let me give you a few examples, but one thing I want to tell you is that super secondary structures can act as the nucleation points in the protein folding. So if a protein is folding, then basically it will fold into super secondary structures, that one alpha helical, then a beta sheet or a alpha helical structure. Let me give you an example to clarify my point. So for example, the first image you are watching here on the left hand side is beta alpha beta alpha beta. Why it is important? It acts as a dinucleotide binding fold. So for example, you want to design a protein which binds to DNA, then you will incorporate beta alpha beta fold so that your protein can bind to a DNA structure. You can see the last one here is a Greek key. Now Greek key is a very stable structure. So if you want to introduce a fold which is extremely stable and you want to stabilize your protein which might not be structurally very stable and it might be heat labile, then you would introduce a Greek key structure. Now, the first one here you are watching is cytochrome P450. There is a particular reason I am giving you this example. One, cytochrome P450 it's the only structure which is alpha helical loop, alpha helical, and then another loop, alpha helical and alpha helix. But you can see that there is a finer structure in here. Let me point it out to you here. So do you see this particular inset? Sometimes proteins do have extra moieties inside it, which we can call cofactors or certain groups. For example, hemoglobin carries heme group, chlorophyll carries a structure. Then you can also see different oxidoreductases have flavin-adenine dinucleotide and several structures like that. So all these prosthetic structures, so protein can carry embedded in its polymer various structures like that. And then after that, the structure you are watching is of an antibody. And how does it look like? Basically, it's a beta sheet, beta sheet, beta sheet, beta sheet, and a beta sheet. But if you have a lot of beta sheets like that, usually a lot of transmembrane proteins have such structures. The N1 is a lactate dehydrogenase. Now, lactate dehydrogenase has a particularly interesting structure, but because it's a dehydrogenase, it also give and take electrons, and it must carry an extra proteinaceous moiety as well, if I can say that. Okay, now the first one here is retinol binding protein. It's pretty much like a beta can. It has this around 8, you can count them, around 8 beta sheets. So if you know about fluorescent proteins, they also have a very similar beta can like structure. If you look at the last one here, which is a triose phosphate isomerase, so it has pretty much like beta sheet alpha helical structure. So up till now you must have noticed that either it's a beta sheet or alpha helical structure and a loop between them. But when you are designing your protein, what you do is that you pick up this whole structure and incorporate it in your new protein. The middle one here is known as a Swiss roll or jelly roll because it looks like that, because there is an alpha helical structure, beta sheet and beta sheet and beta sheet, and it's an anti-parallel beta sheets. So that's why it looks like a Swiss roll. Now, tertiary structure. you have to be very careful because a lot of people confuse that what do we mean by a tertiary structure. Tertiary structure of a protein refers to the arrangement of amino acid side chains in the proteins. Now, if a protein uh, is functional on its own, then that tertiary structure is the final structure. But if it has a lot of subunits, it becomes a quaternary structure. So domains usually consist of two or more layers of secondary structural elements. तो आप काफी जगह सुनेंगे कि हमने ये डोमेन इस प्रोटीन से लेकर यहां लगा दी, इस प्रोटीन से लेकर यहां लगा दी, and that is a domain is composed of two or more super secondary structural elements. And basically when we are talking about a tertiary structure, pretty much एक let's say semi functional or functional entity is formed. एक पूरी polypeptide fold हो चुकी होती है और उसकी हाइड्रोफोबिक कोर एक्वस एनवायरनमेंट से सील हो चुकी होती है, यानी कि उसको पानी कोर तक पानी नहीं पहुंच सकता. नाउ इन दिस डायग्राम यू कैन क्लियरली सी कि एक सेकेंडरी स्ट्रक्चर और सेकेंडरी स्ट्रक्चर, टर्शियरी स्ट्रक्चर इज कंपोज्ड ऑफ अ लॉट ऑफ सेकेंडरी स्ट्रक्चर्स, एंड देन दो टर्शियरी स्ट्रक्चर अगर वो एक फंक्शनल नहीं है और उसको बाकी अपने साथ और फंक्शनल एंटिटी चाहिए तो वो मिलके क्वाटरनरी स्ट्रक्चर बनाएगा. लेट मी गिव यू एन एग्जांपल. हीमोग्लोबिन है, उसके 4 सब यूनिट्स होते हैं, उसमें 4 बेसिकली टर्शरी स्ट्रक्चर बनाने वाली जो चेन्स हैं वो आपस में मिलती हैं तो प्रोटीन फंक्शनल होती है. इफ आई टॉक अबाउट लैक्टेट ऑक्सीडेज, तो समटाइम्स इट इज इवन फंक्शनल विद 8 such subunits. So that makes a quaternary structure, which is the final functional entity. Most domains consist of 40 to 200 amino acid residues and then have an average diameter of approximately 25 angstroms. Many domains are structurally independent entities, therefore are a very important tool for protein design and engineering. हम जो बार बार डोमेन के ऊपर एम्फसाइज करते हैं, वी नो कि अगर हम पूरी की पूरी डोमेन डाल रहे हैं ना, वो फंक्शनल होगी. एक चीज आपके दिमाग में एक क्वेश्चन जरूर आ रहा होगा अगर आप लोग मुझे ध्यान से सुन रहे हैं कि हम तो कोई भी प्रोटीन डालेंगे तो वो एक तरीके से फोल्ड हो जाएगी. एक्चुअली इफ यू लुक एट द थर्मोडायनेमिक्स ऑफ प्रोटीन फोल्डिंग, देन यू वुड अंडरस्टैंड कि एक प्रोटीन कई तरीके से फोल्ड हो सकती है. When we pick a domain, then what we are actually ensuring is कि जो डोमेन है वो उसी तरह फोल्ड हो जिस तरह हम उसको जानते हैं. अदरवाइज इट वुड बी अ जम्बल्ड अप मैश ऑफ अ पॉलीमर जो हमारा, जिससे हम कोई फायदा नहीं उठा सकें. लेकिन हम क्योंकि दीज आर रेगुलर स्ट्रक्चर्स, तो हम इनको क्रिस्टलाइज कर सकते हैं. एंड वंस वी कैन गेट क्रिस्टल स्ट्रक्चर्स, देन वी कैन एक्चुअली सी कि एक एक एटम कहाँ पैक है. देयर कैन बी साइक्लिक सिमेट्रीज, देयर कैन बी डाइहेड्रल सिमेट्रीज and tetrahedral symmetries, octahedral symmetry and icosahedral symmetry. I'm not going to go too much into detail of how the crystal structures are formed and what we can learn from that. If you want to learn more about them, then do consult biochemistry textbooks, as it is more relevant to the people who are studying biochemistry as an independent subject. But one thing where it makes a huge impact is when it comes with the protein folding and selection of the domains, which we will discuss in the next topic.